Hello everyone. In this quick video, I'm going to explain how to complete your lab report based on the potato osmosis lab that you did a few days ago. Now, this poster template you might have already downloaded from Canvas and shared with your lab partner. This poster must be prepared by yourself and your partner, not more than 2 students participating to complete this lab report. Now, the first thing, before you begin working on this poster, first thing that I want you to verify is the poster design page setup. To check that, you go to File and Page Setup. And once this small window pop up, and please make sure that this poster design is on custom setting, where 36 by 24 inches a dimension. And then if it is not, sometimes it may change back to the default standard for 4:3, and make sure that you change it to 36 to 24 in custom format and hit apply, and you will see this like this. Now, when you look at this, you have a couple of areas here. One area here we call introduction or background information, objective and hypothesis, materials used, methodology and procedure, and data and analysis and conclusion, and pictures or references. So these are the areas that you will be completing, along with coming up with a strong descriptive title. And you don't change anything here, and then you just enter your name, complete name, and your partner's name here. And at the very bottom, you just change that year as well. So those are the only thing that you'll be changing, and you don't change any of other standard format. So now you know that it's hard to see when it is in this view, so you are required to bring it zoom in to 100% so that you can see what you're doing. So you'll be working section by section. So in this area, what you will be doing is, first you have to make sure you read at least 3 articles which is given in the Canvas. The same assignment, Potato Osmosis Lab assignment link. Then read it, take notes if necessary, and then you prepare a paragraph summary. So what you're doing here is you're pretty much giving, tell the reader that what is this lab about, and what exactly you're investigating, and what is osmosis, and why are you using potato, and which is our, which is the source of our plant cells, and that's the cells we are using it, and why osmosis happen, and how osmosis happen, and which way water moves, and tonicity, hypotonic, and isotonic concentration. So that pretty much the background information is what you put it in here. And make sure you keep the font size 12 and it is double spaced. And the second part here is the objective, purpose, and hypothesis. So you will get that information from the lab information page, which was attached in the Canvas assignment itself. And you pretty much type a small paragraph here. What is the primary objective of this lab, and what exactly you're trying to investigate. And then you're going to list your hypothesis here. There are 3 hypotheses which we went over in the classroom, so please make sure you put all the 3 here, and a small paragraph explaining the objective and purpose of this experiment. And right here you will be listing all the materials that you use, which includes the culture tubes or test tube stand, and balance, and paper towel. What are anything that you have used for this lab experiment. You will be and sugar solutions, not sugar, salt solutions under different molar concentration, food color, all those things that you will be listing out here. You may list as a 2 column, so it looks better as well. And here you can change that, instead of fall, you can change that to spring 2023. And the next part right here, if you scroll it on the top, is the methodology or the procedure. So what exactly you did in this whole experiment? You just explained that in this paragraph, the whole entire procedure. So how did you cut the potato cylinders, and what did you use to cut the cylinders, and um, and um, what type of potato you used it, and how did you do that, and different type of solutions that you used. And so explain the whole thing in a paragraph form, and do not forget to mention here about your variables you are testing, and what are the constants that you are using here, and if you have any controlled setup here, you mentioned that as well. So that is what you are explaining here in a paragraph form. Now this is the most important part of the experiment, which I'm gonna zoom down a little bit. So here you go, the data. So this is where you're going to place all the data table. So let me quickly show you that. And this is three data table. This is a sample data table anyway. This may not be yours, so I'm just using one of the sample. And this is a sample data table 1. This is the summary data table that you calculated. If you can change all these colors on your own. And this is the second data table, which compares your color and the molarity concentration that you identified. And this is the last data table, which explains the concentration of salt solution and percent change in mass. All the three data table along with the graph. You can easily just copy and paste it right here. Right here in this area, data table and the graph. All the three data table and the graph will go exactly right here. So that is very easy. Just once you already finish working on this, you can just copy and paste there. If you need any help, just let me know. I can help you with that when you do it. So a couple of things that you may have to be a little bit careful when you some formatting error might happen, depends on which computer you work on and how you do this. And if you need any help on that formatting problems, just let me know. And another part of this lab report is the last one right here. This is the most important part of the lab. This is the part that I'm going to read first, which is your analysis and conclusion. So you have to take extreme caution when you type this paragraph. Let me explain what are the things I'm looking for in this analysis and conclusion. So the first one is analysis, second one is conclusion. So in analysis, what you do is you pretty much explain the data that you got here, why the graph is looking like this, and why did you why did you plug in these numbers here, all those kinds of things. So let me go back there again. So in analysis, you're going to explain how did you calculate the percent change in mass, how did you get this total change in mass, and how did you arrange these numbers here based on the color. How did you come to a conclusion that purple is 1 molar and yellow is 0 molar? So how did you do that? So that's what you're going to explain. And same thing here. How did you arrange these numbers in this order here? So you're going to explain that in analysis. So the next part is the graph. So why this graph looks like a negative slope. So all these things you're going to explain it in a paragraph form right here in this analysis box. Let me go back here. The next part is conclusion. Conclusion is nothing but your finding. So remember, we had 3 questions to answer. Those those 3 questions and 3 hypotheses. These 3 hypotheses, whether you accept or reject, and you accept, you would give a reason. Why did you accept it? And one thing I would like to look for is the evidence from the data. For example, one of the question that we are trying to answer was, what is the concentration of potato cytoplasm? So to find out, you will be looking at this graph, and you know that the positive side of the graph is where the potato cylinders gained the mass, and the negative side of the graph is the potato cylinders lost the mass. So in that case, you know that according to osmosis, if the potato cylinders lost the mass, means those cells were surrounded by the hypertonic environment, and this is the positive side. These cylinders were surrounded by the hypotonic environment, and then in between the zero line is the isotonic. So that's why, to find out what is the isotonic environment, which means inside the potato cell and outside the potato cell in the surrounding solution, the concentration of the solutes or the water, it's pretty much the same, which here it represents the zero line. So when you look at this, the line of best fit passes through the zero line at this point. So if you look all the way down to the x-axis, the concentration corresponding to the zero line is coming somewhere around here, which is, for me, I would say it's like around 0.3 molar. So 0.3 molar is the isotonic concentration, which is equal to the potato cell cytoplasm concentration. So that is what you conclude, and you would explain that in your lab report, that the potato cell's cytoplasm concentration is, according to our graph here, is 0.3 molar, because then you have to give a reason, because the line of best fit passed through the zero line and it intersect at 0.3 molar. So you really have to say that 0.3 molar from the graph. The graph indicates that. So when I read it, it should make sense that how did you get that number and from this graph, and how did you generalize that conclusion from the graph? Those are extremely important. That's what I will be looking for, analysis and conclusion. And the same thing you apply to the other two questions as well. Another thing is right here, the last one, the pictures and the references. Whatever the three articles, or if any other resources that you used, and which you are going to give link here. If you know APA format or MLA format, please, you're more than welcome to generate the citation here. That's perfectly fine. Minimum 3 I'm looking for, and then probably some relevant pictures here for the test tube pictures and those potato cylinders pictures. So this is what we are looking at, and if you have any particular questions, please let me know. Email me or send me the message in the comment when you're preparing it. I'll help you as possible. And then one last thing is the title of your experiment. What titles I do not want? I do not want Potato Lab, Osmosis Lab, and I don't want these type of title, or I don't want to see that, okay, Salt Solution Lab. Those type of titles I do not want. If you look at the rubric, you will see that the title should be descriptive. So when I say descriptive, it should tell the reader what investigation you are doing. So you can say, one of the example is like calculating the concentration of potato cytoplasm, or that's or an experiment to prove or to show finding out the potato cytoplasm concentration, or you can say an investigation to find out the percentage change in mass while potato cylinders are placed in different salt concentration. So those type of descriptive title, that is what we are looking at. Now always remember, language matters, the structure of the language, sentences matters, spelling matters, all those things. Proofread, and you can split the work, and half of the work you can do it, other half your partner can do it. So remember, later you will be working on this poster all by yourself in the next unit. So please make sure that you work diligently on it. So another important thing is the font size. I do not want the font size more than 12 and typing anywhere of this information. And then once you complete with the whole thing, and make sure you download this file, export it as PDF, and then you submit in the Canvas. So I hope this video give you a kind of idea on how to find this, how to write this report. And if you have any question, please email me or ask me in the classroom. So thank you very much.